The students, today will discuss about protoplast isolation, purification, culture and fusion. Protoplasts are plant cells without a cell wall. Hastings used for the first time the term protoplast in 1880 to describe living matter which was enclosed only by plasma membrane. The plasma membrane now acts as barrier between the external environment and interior living parts of the cell. Protoplasts, because of absence of rigid cell wall, can be effectively served as recipient hosts for transformation of recombinant DNA and proteins. They can also be used to study molecular architecture of plant cells. Economically important somatic hybrids can also be produced by fusing protoplasts from genetically diverse plant species. The isolation of protoplasts from plants can be done either by mechanical disruption or enzymatic degradation of rigid outermost cell wall. Generally, tissues of young leaves are used as primary source of protoplast isolation, as they release healthy protoplasts with highest cell viability. Now, let us take up protoplast isolation. Protoplasts from plant tissues can be isolated by employing either mechanical or enzymatic methods. First, let us take up mechanical method. The cells are first plasmolyzed by subjecting to plasmolyticum treatment. This leads to the outflow of fluid from interior portion of cell, resulting in the withdrawal of cell contents into the center. Plasmolyzed tissues are then spliced into several small pieces, followed by deplasmolization, which results in the release of protoplasts. In this method, the protoplast sources are usually soft parenchymatous tissues which can be easily separated and crushed into pieces mechanically. This method is not commonly employed as viable protoplast yield is normally low, but from being tedious and laborious. Now, after this, let us take up enzymatic method. The hydrolytic enzymes which can degrade the rigid cell wall of plant cells are used to isolate the protoplast. Using this method, protoplasts are isolated from leaves, pseudapices, stem and embryos. However, the mesophyll tissues of young leaves are the most suitable source of protoplast isolation, as they can be easily assessed by cell wall degrading enzymes because of their loose and spacious arrangement. Cellulase, hemicellulase and pectinase are used to remove the cell wall from the plant cell. Pectinase degrades the middle lamella, while cellulase and hemicellulase digest the cellulosic and hemicellulosic components of the cell wall respectively. When leaves are used to isolate protoplasts, they are first surface sterilized using appropriate surface sterilizing agents. The lower epidermis of sterilized leaves is peeled off using forceps, followed by chopping the leaves into pieces with sharp scalpels. The leaf pieces are suspended in enzyme mixture of pectinase or macroenzyme and cellulase with pH adjusted between 4.7 and 8 and temperature at 25 to 30 degrees Celsius. Sugar alcohol, sorbitol or mannitol are usually added as osmoticum to the solution to stabilize the fragile naked protoplast and to prevent from bursting. The naked protoplasts devoid of cell wall are released gradually in the enzyme mixtures, which are then washed in a suitable washing medium to remove enzymes and the debris. Now, after this, let us take up protoplast purification. The suspension obtained after enzyme treatment consists of released naked protoplast, undigested tissues, and cellulose debris. Purification involves passing of protoplast suspension through nylon or metal sieves of decreasing pore size to remove the larger unwanted materials. This is followed by gentle centrifugation to pellet the protoplast. Centrifugation and resuspension of the pelleted protoplast may be repeated several times until a pure suspension of protoplast is obtained. Next, let us take up determination of protoplast viability and efficiency of cell wall removal. The isolated protoplasts have spherical shape when observed under light microscope. The staining of protoplasts with fluorescent brightener tinapol will produce yellow fluorescence if there is any remaining cell wall. The viability of protoplasts can be determined by using fluorescent diacetate, in short form FDA, which passes across the plasma membrane of cells. The viable protoplast will produce fluorescence of green or yellow color, while the dead protoplast will not produce any fluorescence. Let us take up protoplast culture. Protoplasts are cultured either in liquid or agar solidified media. Culture media prepared according to the formulations of Morashi's and Skook, Gambok and cow are used extensively for protoplast culture. However, as protoplasts are extremely delicate in nature due to absence of cell wall, there is a need to slightly modify the original formulation of normal culture medium. For example, the ammonium ion concentration should be reduced in protoplast culture medium, as they are detrimental to protoplast survival. Maintaining appropriate osmotic pressure is also important, as newly isolated protoplasts are osmotically fragile and are susceptible to osmotic lysis. Protoplasts undergo repeat cell wall synthesis immediately upon removal from enzyme solution. It may take few days to complete cell wall synthesis under suitable condition, and protoplast lose its characteristic spherical shape, indicating cell wall synthesis. Now, let us take first the liquid culture. The protoplasts are suspended in liquid culture medium at required plating density and dispensed into appropriately sized petri dishes. The cultures are sealed with gas permeable tape and incubate in growth room at about 25 degrees Celsius under low intensity illumination. Next, let us discuss liquid droplet method. The protoplasts are suspended in liquid culture medium and droplets are pipetted into smaller size petri dishes. The plates with 5 to 7 drops are sealed and incubated. This method is convenient for microscopic examination, but one of the limitations is the clumping of cultured protoplasts at the center of the droplets. Now we have the hanging droplet method. This method involves the placing of small droplets of protoplast suspension onto the inner side of the lid of petri dish. When the lid covers the petri dish, the droplets are hanging down from the surface of the lid. Pure protoplast or droplet can be grown using this method as compared to the conventional ones. Next, we have the feeder layer method. In this method, the X-irradiated, non-dividing living protoplasts are plated onto the agar solidified medium to form the feeder layer. The protoplasts are plated on these feeder cells at low density. The feeder cells provide the necessary growth requirement for the isolated protoplast. Now we have the co-culturing method. This involves the culturing together of slow and fast growing protoplasts. The fast growing protoplasts produce growth factors and chemicals, nutrients necessary for the cell wall growth and cell division of the slow growing protoplasts. Now let us discuss protoplast fusion. Isolated protoplasts, when they come in close contact, may get fused spontaneously or may be induced to fuse by using fusogens. Fusogens are fusion-inducing agents which, when applied to the protoplast mixture, will produce fusion products. Now let us discuss spontaneous fusion. The cells which are in close contact may fuse together spontaneously during the enzymatic degradation of cell wall to form homokaryons, the cells having two similar nuclei. Most of the time, multinucleate cells are observed immediately after enzymatic protoplast isolation. However, the protoplasts do not fuse spontaneously once they are freely isolated. The spontaneous fusion products in most of the cases do not give rise to whole plants, as cell division continues only for a few cycles. Let us take up induced fusion. Under induced fusion, we will first take up sodium nitrate treatment. The fusion of protoplasts from two different plant sources is performed under the influence of sodium nitrate as fusion-inducing agent. The protoplast mixture is suspended in 10.2% sucrose solution containing 5.5% sodium nitrate. The suspended protoplasts are incubated in water bath at 35 degrees Celsius for about 5 minutes, which is then followed by centrifugation at 200 G for 5 minutes. The protoplasts are fused during the time of incubation. Next, we will discuss high pH and high calcium ion treatment. The protoplast mixture is subjected to high pH around 10.5 and high calcium ion concentration around 50 millimolar per liter at 37 degrees Celsius for about 30 to 35 minutes. The protoplasts undergo fusion under this condition, and about 40 to 60% of protoplasts fuse to produce fusion products. Let us discuss polyethylene glycol induced protoplast fusion. This method is most commonly used as it produces high frequency fusion accompanied with low toxicity to most cell types. The isolated protoplasts are first treated with 28 to 50% PEG for about 15 to 30 minutes. This is followed by gradual washing of protoplast to remove PEG from the surface of the protoplast. The protoplasts are fused while washing in the medium having high pH, which is around 9 to 10, and high calcium ion concentration, again which is around 50 millimolar per liter. Now let us take up electrofusion technique. In this approach, the protoplasts are made to fuse under the influence of electric pulse. The protoplasts are first brought to close contact by using low voltage, non-uniform alternating current pulses. The fusion of the contact protoplast is brought about by a short pulse of high voltage of about 500 to 1000 volt per centimeter for few microseconds. This leads to the breakdown of membranes where the adjacent cells are in contact, resulting to the fusion of protoplasts. The entire operation is carried out manually in specially designed equipment called electroporator. In this method, there is no control on the type of protoplasts which are fused. A slight modification to this technique leads to the development of microelectrofusion method. In this method, the protoplast pairs are individually transferred into the microfusion chamber, and each microfusion chamber containing one pair of protoplasts are induced to fuse by electric current. This technique produces high specific and nearly 100% pairwise fusion. The electrofusion method is simple, quick, and efficient, and does not produce any cytotoxic response, but requires highly specialized device to accept cell fusion. Now, after discussing all these things, let us come to the conclusion. Protoplast isolation and successful culture of isolated protoplasts are important steps towards production of useful somatic hybrids. The enzymatic methods of protoplast isolation using cell wall degrading hydrolytic enzymes are more effective than the mechanical methods. The isolated protoplasts can be cultured by adopting different culture methods. Feeder layer and liquid droplet methods are considered as more effective than other culture methods. The isolated protoplasts can be made to fuse either by spontaneous or induced fusion method, which uses a fusion inducing agent called fusogen. Now, combination of high pH and high calcium ions along with PEG treatment produce high frequency fusion products, which can be further raised to somatic hybrids through subsequent culture in suitable medium. The somatic hybrids generated can then be integrated successfully into the breeding program for generation of improved crop varieties.